Hi guys, welcome back to another video, and in this video we will today cover cell junctions, which are of three kinds: occluding, anchoring and communicating. So when I talk about cell junctions, I would first like to introduce you to the cells, individual cells. Now the cells together are responsible for the functioning of multicellular organism, and each cell is linked to its extracellular matrix and another cells through their cell junction, forming the tissues. These cell junctions are classified into three functional groups: occluding, anchoring and communicating type. So when we talk about the occluding type, that indicates stop. Occlude means to stop. That means it is forming a barrier and therefore sealing the cells together. Hence, there are no gaps and a barrier is formed. It is further divided into tight and septate junctions. Next type is the anchoring type. Anchoring, that indicates to attach. So cells are mechanically trying to attach to the neighboring cells or the extracellular matrix surrounding them. Thereby they are holding the cells together. They are further divided into adherents or desmosomes. Now the third kind, that is the communicating kind, indicates that there is an interaction of cells involved. That means the two cells are trying to either mediate the passage of signals, which could be chemical or electrical. Thereby the cells are interacting together, hence through the communicating junction, which is further divided into two kinds: gap junction and plasmodesmata. Now let us check these in a little further details. So when I talk about the occluding junctions, they seal the cells together so that it forms like a barrier. So when we talk about these, they are further into tight and septate junctions. The tight ones are also called as zonular occludins, and they are a cell-to-cell junction. Now, transmembrane proteins here play a major role in these junctions as a communicating proteins, and they are further interacting with intracellular peripheral membrane proteins called as zooproteins, which I will depict in a picture here. So yeah, here's the picture. Between these two cells are the tight junctions, which are kind of like forming a barrier for these solutes to not pass across this tissue. So hence this is a barrier kind of thing that is being formed by tight junctions. And when we talk about an individual cell tight junction, so these are the transmembrane proteins, which could be claudin or occludin, and these further interact with or associate with like intracellular peripheral membrane proteins, which are called as zooproteins. Zooproteins further are linked or trying to stabilize this junction by interacting with actin filaments. Septate junctions are the main kind of occluding junctions in invertebrates. Now, anchoring junctions are responsible for a cell-to-cell or a cell to extracellular matrix attachment, and they are present in animal cells or animal tissues. Now, there are two kinds: adhering and desmosomes. When I talk about adhering junctions, could be further seen of two kinds, because there's a cell to cell and extracellular matrix interaction. So when I talk about the cell to cell interaction, it is called as adhesion belt, and with extracellular it is called a plaque, however you pronounce it. And then cadherin and integrins are the kind of proteins that are involved in either of the two. Then when I talk about the cell cell interaction here, I'm trying to depict this diagrammatically, or extracellular matrix interaction with the cell. Here's the presence of these transmembrane proteins, cadherins or integrins. Now remember, these transmembrane proteins are protruding out of the plasma membrane, and they are also further attached to the adapter proteins, which are present inside the cell, intracellular. And to stabilize this entire arrangement, there's an interaction of adapter with actin in case of this entire type, that is the adherent junction type. So actin is involved in the adhering junctions. Again, when we talk about desmosomes, again cell to cell and cell to extracellular matrix. If it is cell to cell, then it is called spot desmosome. If with extracellular matrix, it is called as half desmosome. And further, cadherin and integrin again are involved in either of the two. When I talk about the cell and cell interacting here, in case of desmosomes, the presence of these cadherin integrin proteins, transmembrane proteins, again further interaction with the adapter proteins, but here instead of actin, intermediate filaments are responsible for. Third kind is the communicating junctions, and this is between cell to cell, but it is present both in animals and plants. In case of animals, it is called gap junction. In case of plants, it is called plasmodesmata. Gap junctions are like gated channels because they are linking the cytoplasm of the two communicating cells, and plasmodesmata are more like open channels. Since these are gated channels, pH and calcium ions are also involved. Plant cells which are adjacent to each other communicate through cytoplasmic connections which are called plasmodesmata. They are analogous to the cell gap junctions. More tubule is present between the two cytosols, which is actually an extension of smooth endoplasmic reticulum. Now, gap junctions have transmembrane proteins which are called as connexins. They allow the passage of intracellular signaling molecules like cyclic AMPs, calciums between the adjacent cells. 6 connexins form one connexon and 2 connexons form one gap junction. I hope you learners found this small video helpful. Thanks for watching.